this session is mostly about kind of presenting the groundwork and what we know or think we know about this area. It's going to focus a lot on the lower gut, because that's where most of this data we have is. But as I'm told by my kids, what I do is primarily a mixture of electrics and plumbing, in terms of the fact that the gut is a tube. It's, from your mouth down to your anus, it's one long tube, which has essentially a nutritional and a digestive and excretory function. And that plumbing part is plainly neurally controlled. And what I'd like to persuade you of is that there is enough knowledge out there on which to base best practice principles. Not to be remotely autobiographical, but just to say my backstory, because it's relevant to how I present this stuff, is that I was originally a neurologist who then became a gastroenterologist by mistake during my research. And I work both at that building on the left and the more attractive building on the right, in a very lovely city which has been my home. So if we talk about the colon, first of all, in terms of function, it's the paradigm for talking about digestive function as well, because I hope I persuade you in the next 2 or 3 minutes that understanding colonic motility and colonic barrier function is very akin to understanding what happens in the upper gut. So the critical issue, of course, as you know, the cliche is this: you have voluntary control of what you eat, up to the point you swallow it. Thereafter, everything that happens digestively is completely autonomic, until it comes down to your rectum, at which point you have voluntary control of whether you... And so you have these two reservoirs. You have a reservoir in your stomach and a reservoir in your rectum, and everything in between is out of your voluntary control, yet it is controlled. So in spinal injured patients, who are my major patient group nowadays, there is still some preservation of function, independent of a complete spinal cord injury. So there is something autonomic within the gut that regulates all this. Yet notwithstanding that, the control aspect of this so-called brain-gut axis, is the kind of the core theory around this. And for us, we've always thought about this as being a one-way directional system, that something begins here and controls here, that I decide when I swallow, I decide when I go to the toilet, and that's about it. But obviously it doesn't take a lot of intelligence to realize that it must be bidirectional. How do you know when you're hungry? It isn't just because it's 2 o'clock in the afternoon, I haven't eaten. It isn't just because it's 8 o'clock in the morning, I haven't since yesterday. It's something, there's a sensation part. This is a two-way street, and this whole bidirectional part is what, this motor and sensory component, is why a neurologist would be vaguely interested in gut function. And what's emerged even more recently than that notion of sensory function, which is fairly obvious, is the idea that this may not just be driven by the things you are born with, i.e. your neural control. It may be influenced massively by the gut bacteria. And I will talk very tangentially around that, because my interest in this is mostly in terms of one of our fellows running a fecal microbial transplant service project. But a subsequent speaker, Nick Sharma, will talk about this much more, again, from a position of knowledge. So just to start off with a few kind of very mundane things, it's worth remembering that there's about 10 litres of liquid slushing around your intestine in a 24-hour period. You've got your couple of litres you drink, plus a lot of saliva, a lot of pancreatic juice, a lot of gastric fluid. The vast majority of that fluid, about 7-8 litres, is absorbed from your small intestine, and your colon desiccates the last 2 litres. So what comes out of your bottom end is about 100 mils of liquid in the average British stool weight. Interesting concept, being about 150 grams, 2/3 of that is water, so you get about 100 mils of water out of your bottom end, out of the 10 litres that sloshes around your gut. Essentially, therefore, there's this system of extreme control. You have a system where you've taken 10 litres and you've reduced that to 0.1 litre through gut transit. And that is a phenomenon which is mediated incredibly tightly by the nervous system. So we know from denervated animals which have had their autonomic nervous or enteric nervous system destroyed, that ability to fluid retain is completely lost. This is something which is mediated by your nervous system. It's not just a question of transiting through the gut and it just gradually getting absorbed. There's a nervous control to that, and if you destroy your nervous system, you'll tend to have a failure of that system, because your gut is mediating absorption and secretion and movement. So I'm sorry for this, but it's sort of germane to what I'm trying to persuade you of. We think about the gut as being this, obviously stuff flowing through your mucosa there, and you have the kind of typical three-layer structure of a mucosal layer, a muscular layer, and the serosal layer. And what you are persuaded of, I hope, is that your gut, as you know, is the second most neurologically rich organ of your body. It's got more nerves than your spinal cord. Okay, there are 100 million nerves in your gut, and most of those are independent autonomic fibers with very little central relay. But there are some. The point being that peristalsis occurs even in the absence of central control. So the brain-gut axis is about control rather than about function. The function will occur, but whether you're able to control it and lead a good life will depend on your brain being intact. So this is very simple at this level. I'll try and get a bit more complicated as time goes on because it's relevant to where we may move. And what's happened is, for the first time, we've known about peristalsis since Victorian times, since 1840, but what we haven't known is what mediates peristalsis. And at last we know about 30 years ago. And what we know is this. The critical thing that mediates peristalsis is this thing here. They're called IPANs, intestinal panafferent neurons. They're sensory nerves in your gut. And it's worth thinking about them for a split second, if I may. Imagine your gut being this thing where you have your mouth at that end on Borough High Street and your anus there. It's a nice thought for you. And what you have throughout your gut is arrays of these nerves, literally millions of these sensory nerves arranged perpendicular to that tube. So I am the sensory neuron for a second. And when you make that first swallow that I mentioned, that mechanical bolus stimulates these neurons at this end, and via a local mechanism which involves the serotonin receptor 5-HT4 receptor, you get an upstream excitatory contraction, a cholinergic contraction, which squeezes that bolus harder, and a downstream relaxation, so that there's a pressure gradient which moves that bolus along to the next neuron, which then picks that movement up, and so on. And in your gullet, that 40 centimeters is traversed in 5 seconds. In your colon, that 120 centimeters is traversed in 20 hours. So there's a differential ability to do this based on how those sensory neurons interact with each other. Some of them are very swift relays, some of them more dawdly, some of them go backwards. And that is the basis of all this. So peristalsis is now got something we can target. Rather than being this mysterious mechanical phenomenon which happens in the gut, it's a sensory neurological phenomenon that triggers that. Okay, so this is constant interplay. For years I was just a plumber. I just used to put tubes up people or down people and fiddle with things and put stents in and bypass things. But actually, I've stopped doing all that stuff now, thank God. But what we understand much more is that a lot of the control stuff and the stuff that goes wrong with neurological disease is that there's a reason for this. Okay, it isn't some mysterious, because they're immobile, because they're sitting in a chair all their time, they're eating. There's more to it than that, and it begins with some of the physiology, which is the reason for telling you all this stuff in such boring detail. And that has a net effect. The net effect is this. This is, those of us who are in this room are healthy, happy, social, extrovert people. This is how our gut works. This is one of my research fellows, lucky fella, who had the privilege of having me put a tube up his bottom and leave it in there for 24 hours, between 2 o'clock one day and 2 o'clock the next day. And that lucky fella had the tube, which had multiple pressure points throughout it, and it measured pressure in different regions of his gut, from his cecum down to his anus. And you can see nice pressure curves. Some of these things, basically, you get big pressure waves, which begin sometimes in your right-hand side and stop halfway down, or begin halfway down and come further down. And they're busy after lunch, after dinner. Goes to sleep when he goes to sleep, and when he wakes up. You get these flurry of green hills of peristaltic activity through the colon. And you can see what happens to somebody who's got surgery. As it happens, this is one of our MS patients from Queen Square. This could not look more different. Of course, I'm showing you the best and worst traces. And there's almost no peristalsis here. There's almost no green hills here. There's lots of retrograde activity, even at night time. So whereas when you go to sleep, you're very rarely bothered, unless you've got some other functional problem, you're very rarely bothered by nighttime belly cramps or needing to open your bowels. But our patients will tell us, I can't rest at night, something happens. Oh, it's in your head, don't worry, your bowels are asleep when you're asleep. Well, it isn't. When they're asleep, their bowels are work backwards. But it isn't just about transit. I don't want to leave you this idea that everything I'm talking about is movement and transit. There is a huge control aspect to this. So this is a funny back to front graph, but I wanted to try and persuade you how this system, when it comes down to this tank at the bottom, your rectum, how that system coordinates with your brain. This is you and I. This is a healthy person at time zero. And time zero, which is rather bizarrely on the right-hand side of the curve, is the moment when you get off the pan. I never know what word to use for defecation, because defecation sounds so clinical. So I don't want to use any other word. So dump is where I settle for, hope you don't mind. And in the half an hour before you take a dump, you have lots of these green hills. Lots and lots of these big propagated contractions, those big green contractions. Whereas a patient has none of those. Okay, so this is some work done by Phil Dinning, one of our previous fellows. And when a patient with a neurological disease goes to the toilet because their carer has come in, or because it's the time of day when they try to do it every day or whatever, they don't have the benefit of that. So transit is not just about a phenomenon in isolation. Transit is about transit down to the rectum, at which point you can evacuate. We are lucky. We have an intact nervous system which allows us to know when we are adequately full, rather than just having to take a dump when we are made to. And we can demonstrate that radiologically as well. And I've made that point about autonomic control of this more than enough times now, and the critical role of serotonin in terms of potentially treatment targets. As I said, I'm not going to talk about treatments today because it's just about laying the groundwork for what we know. And this is just kind of an iterative slide. And I think the slides are sort of available, and there's nothing secret here, and I'm very happy for anyone to use them. But so don't feel obliged to write any of this nonsense down, but it's there if you want to ever refer to it. There's this issue, the fact that there is this bidirectional system here, where there's a conscious aspect of this, which is the behavioral part about when you use and don't use the toilet, and when you eat and when you don't eat. And there's the unconscious part, which is about, stressors having a role. So those of you who have ever been through periods of stress and your tummy cramps more and you don't go to use the bathroom or use the bathroom too often, there's a logical reason for that. That's through this autonomic system. And the last bit of this, which I think is relevant to all this, because I do want to keep bringing up this analogy between the rectum and the stomach. We don't have enough data about the stomach. We have tons of data about the rectum. But the systems are similar. They are both, the rest of the gut is tubing, but the rectum and the stomach, other way around, the stomach and the rectum are basically tanks. They're capacitance vessels which fill up, this one voluntarily, that one involuntarily, and then at a fixed rate from the stomach, empties, and at a conscious rate from there, empties. So there's an analogy for that. But the other thing that's relevant to all this is in the colon, the idea of the sigmoid colon just above that being important as a reservoir as well. It isn't just the rectum. And the rectum actually seems to be more about sensation. This is some work done by Pratish Trivedi in our group. The rectum is important as a sensor. It's the kind of traffic light system, whereas the sigmoid colon is actually an extended part of the rectum, as the tank, in fact, is what's emerging. So yes, that's making that point that this is too dull to go through, but just persuade you about the headlines. The headlines are what's interesting here. The rectum is what tells your brain what to do and then allows it to do it. Your sigmoid is where the stuff is held up. And shown visually, and there's a point about this because this is related to stomach function. I was sitting on a plane to Gothenburg a couple of weeks ago and had nothing else to do after I'd finished reading what I had to read, so I produced this, so bear with me. I'm not that excited about it. But it is to try and make the point that if there's a takeaway from this so far, I don't want you to think about constipation and problems with defecation as being opposite things to incontinence. They're a spectrum. Right now, you're all sitting here, inverted commas, constipated. You're not opening your bowels right now, I trust. Yet you will voluntarily at some point open your bowels, and you are, inverted commas, incontinent in a toilet. So there's a kind of sliding scale between this. And we mustn't think about our neurological patients as being, oh, he's a constipated one, she's an incontinent one. We slide, and a lot of our neurological patients are absolutely the epitome of this, that they are terribly constipated. We give them billions of laxatives, and suddenly themselves. And that's the issue. It's not a separate problem. It's not necessarily iatrogenic. It's part of the disease, and failure to understand that is quite a big deal. And the point about this is that you have this transit through the gut, which moves along at its whatever rate it's moving along at, getting this volume of the stools gets smaller and smaller. How much time I had on that plane. And this keeps happening through the day. And gradually more and more of that fills, and your rectum, as that sensor stays in a red light position, more and more fills with it. And at a critical point of filling, you get something which then says, oh, we better find a bathroom at some point. Finish the end of the talk and then you'll get there. And at that point, you're vaguely aware of something, but you can suppress it. You can suppress it. If you lose some of that CNS control, you can't suppress it. At that point where you should be in the tank phase, you suddenly switch to the emptying phase at an inappropriate time. And that is distressing. And at that point you then void. Hooray, look at that. So, thus far, you have been persuaded that there is this two-way communication between brain and gut, I hope. And that whilst most of the physiological measurements we make so far are lower GI because we can reach it with tubes, the upper GI stuff is ripe for plucking. There's data to be had there. Plus, there's clinical observational stuff that you will know of. But what's clear, of course, is that there are psychological and psychiatric factors that influence this. And of course, that relates to eating as much as to bowel evacuation. And there are aspects of disease, CNS disease, which influence this as well. And through this axis, which is primarily neurological, but also involves hormones, which I've tried to skip talking about today, there are alterations in the end organ. And it's worth mentioning that one line on gut permeability. So for a lot of gastroenterologists, the leaky gut has been a sort of a dirty word. It's not been acceptable to believe in that. But, the other side of the coin is that in physiology, we absolutely measure in research gut permeability. We do it all the time. And we know that there are situations, including neurological diseases, where there is grossly abnormal permeability. So it's well known that MS patients have a grossly permeable gut compared to age and sex-matched controls. Parkinson's patients, as I'll show you some data later on, have different permeability. The controversy has been around patients with inverted commas IBS and whether that so-called leaky gut has been a problem. And of course, there are charlatans in my profession and in your profession who have exploited this nonsense into believing that there's toxins constantly flowing through our body and that we need to detoxify and all this nonsense. And that's a danger. But there is a true phenomenon of gut permeability which I hope is resurrected in a healthy way. So I think one of the things you'll see as you keep abreast of this field going forward is that this concept will get cleaned up and re-enter the science and be a therapeutically useful thing, especially in talking about potentially the microbiome amongst other things. And all that results in outcomes that we care about as clinicians. I absolutely don't want to talk about environmental factors and genes because I don't think that's, from my perspective, it's the least interesting part of what we can change. We, by and large, I'm very unsuccessful in changing people's behaviors in terms of other things, and I certainly can't change their genes. And so I, by and large, mention it as something for completeness rather than something I necessarily want to persuade you is the critical part of what we want to do. But the main reason for showing this figure is just that it illustrates the idea that this thing that we focus on today is one part of something upstream and something downstream clinically. And the bulk of this rest of this talk I'll be sort of subjecting you to now is around the idea of how we'll present these concepts, the idea of the fact that there's a psychiatric comorbidity with functional disorders, that tricyclics help people with IBS, that there's an overlap in the serotonin receptors and receptors in the brain and the gut, and that there are stressful things which alter gut function. So there's this huge issue around both dyspepsia and IBS and the brain, which is relevant to understanding neurological disease. So transit I've mentioned, and I won't mention again other than to just remind you that there's this rapid transit through there, a couple of hours spent in the stomach, about a couple of hours spent going through the small bowel. This kind of 6 meters of small bowel takes 2 hours, and then the next 20 hours in health is spent in the colon. So the 80-90% of your oro-anal transit time is spent in your colon. Okay, that's well known to all of you, and the relevance of that will become clear as we go through. Because if we work our way down the gut, and think about neurological diseases, there are a few things to help, I think, understand. So those of you who look after PD patients, there is a reason why they... So one of the things we've learned recently is the issue around why some people have this famously on-off effect with their medication, and that is incredibly closely related to their L-DOPA kinetics in their bloodstream. And what we know from some really nice data is that if you look at patients with Parkinson's, roughly half of them have a normal profile of their L-DOPA, and about half don't. And in that half who have a normal profile, the majority of them have normal gastric emptying. This phenomenon of roughly 2 hours after you eat, stuff emptying into your intestine is there in about 2/3, but 1/3 don't. So somehow it's getting into their system despite delayed emptying. Whereas amongst those with delayed, who are the ones who get much more on-off effects in terms of their medication, the vast majority have abnormal emptying. So abnormal gastric emptying is critical to a lot of, not all, but to a lot of Parkinson's patients' response to treatment, because they have, and that abnormal gastric emptying is even before, from data we've got, even before they have their medications. And one of the paradoxes is that the medications we give them for their Parkinson's makes their gut slower, rather unhelpfully. But even before that happens, they have this abnormal gastric emptying. So the whole thing about appetite control and interest in satiety is going to be rated, apart from the medication effects, there's obviously going to be a more general eating behavioral aspect to this in terms of the motility through that region of the gut. And the opportunity that comes with this, and I said I wouldn't talk about treatments, but I'm going to, is that there are drugs like prucalopride, not the, but the serotonin 5-HT4 drug, I mentioned 5-HT4 at the bottom of those sensory receptors, which can accelerate gastric emptying in some people, and therefore turn the kinetics, the L-DOPA kinetics, and therefore improve clinical outcomes. So you have that structure in mind of the brain, the gut, symptoms from that previous slide. We can try and influence something there to influence something down there in terms of outcomes. We are very much in the kind of pygmy stage, in the infancy of understanding all this stuff. So if I'm presenting this, the reason I've almost deliberately not presented clinical data is because there isn't a lot there as yet, but it's an area which is ripe. I said all that nonsense already. The other aspect of gastric emptying, which is relevant to a lot of practice, is the whole idea of diabetic neuropathy. And all of you are familiar with this kind of concept of diabetic gastroparesis. And it's a real phenomenon, it's a real thing. It's about the fourth biggest cause of mortality amongst type 1 diabetics is aspiration. So it's surprising. We tend to think people don't die of aspiration anymore, but in DKA, the second commonest cause of death from DKA is not metabolic stuff, it's aspiration. And this is complex. It's complex in the sense that gastric emptying is altered by the level of sugar. So the higher your blood sugar, the more abnormal your gastric emptying. But also the diabetic neuropaths are the ones who get abnormal gastric emptying. It isn't a phenomenon of all diabetics. It's only the ones who have an autonomic neuropathy, in particular in relation to nitric oxide and to the cells of Cajal. So there are markers. I don't want to get too boring about this, mostly because I don't understand it, but there are markers of what can mark that, so the neural networks that are abnormal in diabetic patients, which influence gastric emptying. And there will be drugs. There are so-called tachykinin drugs being developed to target this. There are nitric oxide donors being developed, which may have an effect on gastric emptying. There are drugs in evolution which may help. I'm not by any means suggesting that drugs are the solution to this problem for everyone or even for anyone, but they're just the fact that as we open targets up, drugs open up the discussion and open up us doing things better. And most of what we do to look after patients with diabetic gastroparesis is actually just to feed them carefully post-pyloric, but there are ways of adjuvantly helping that along the way potentially. And I'm slightly obsessed by this. I think there's a real issue around this. We tend to think about functional disorders as being the only patient group who have complex past histories. It's an interesting phenomenon. Everyone says about IBS. Well, IBS patients are a bit strange, and they always quote this figure saying, well, 35% of them have been victims of child physical or sexual abuse. But what's incredibly interesting, and that's true, no one's denying that, and that's usually from tertiary centres, so it's not everyone, that's kind of the population group of IBS who end up in a specialist centre. But if you look at the same tertiary centres' incidence of childhood physical and sexual abuse amongst their Crohn's and colitis patients, guess what that proportion is? Think of a number. The number is 25%. Now, no one says, oh, well, abuse is what causes their Crohn's. Okay? But it's an interesting phenomenon about why that may be. And I think it relates to this. I don't often read the primate literature, but there's an interesting primate paper here looking at apes and apes who have been abused. And ape abuse apparently is quite a big thing, not by humans, by apes abusing apes. And what you have is control animals which have been healthy, animals which have been abused, and animals which have been abused and in maturity became abusive. And what you see if you're looking at their serotonin kinetics in their brain, measured as a substitute by CSF metabolites, is that there's a very big difference in this. So there's stuff which happens cortically which influence sensitivity in the gut, and that is probably critical to the evolution of this. And there are nice, interesting theories that if you take this model and give it a very low-grade stressor, you can make some of these animals into animals which have a form of Crohn's disease and colitis. Okay, so there's some interplay between these horrible things that happen to our patients in young life and environmental triggers which in otherwise healthy people would be ignorable. So this idea of brain and gut is not just about going to the loo or not going to the loo, it's also potentially about some of the inflammatory diseases. And some of the stuff that you'll hear from Nikhil in a while, and which I'll move to towards the end of this ramble, is the idea that sometimes there may actually be stuff which begins here and goes up here, causes disease that we think is initially primary neurology, but actually some of these primary neurological diseases may begin in the gut, from gut permeability, going up here, altering kinetics here, and then coming back downstairs and giving us side effects of conditions. So there's an interesting phenomenon that's worth telling here, if you're happy for me to go through that story. And it's worth thinking about stress because, again, the whole stress effect in gut function is a mimic and a model of how brain diseases affect the bowel. Sorry to present, I don't do any animal work, so I'm presenting other people's data here. But if you take an animal model of stress, which is when you separate a pup from its mother, you can get these maternally separated animals and normal animals, and you see that the maternally separated animals have diarrhea. There's an effect of this acute stress on these rat pups, and that relates to something that happens in their gut. In other words, the point about this is that you can do something to somebody here and change stuff here. Now that's pretty obvious to all of us who've got kind of stress-induced diarrhea before exams or butterflies in your tummy before your wedding day or whatever else. But it's not just the symptoms that change. There are hard and fast things like the number of goblet cells in your gut that change. Could you anticipate that? You just think, oh, it's all wiring, but it isn't just wiring. There's other structural stuff that happens in your gut. Your gut is basically, I think, a gigantic window of stuff that happens. It isn't a disease organ in its own right necessarily. Sometimes it's just a window of stuff that's happening upstairs. And we can show this in other ways. This is again data from that same Japanese group before, showing that this whole mechanism occurs through a sympathetic nerve pathway. And here's us people putting balloons into people's rectums. This is stuff done with Joel Winston at the National, showing that you can get people getting more excitatory messages from their gut if you've stressed them beforehand, and you can then block that with an antibody to the sympathetic nervous system, which then makes everything go back south. So this is a sympathetic nerve mediated phenomenon. This sensation of how you sense stuff here and goes up there and then comes back down. That's the goblet cell stuff before, but you can block that in the first place by blocking the antibodies with antibodies here, so that you can interfere with that, and that's a potential target for the future. So this is, I spend my life sitting with with immunologists who talk about inflammatory bowel disease, and they have these complex figures with leukocytes migrating there and cells coming out of there and cytokines being produced there, and it's magnificent. This is the best I can do. This is where we are. We know that stresses occur and are mediated by central structures, that there is something, I've tried to present this in a patchwork through the data I've shown you so far, that there's descending pathways which act on the dorsal horn, which then result in some sort of sensitization, so that when there are simple insults in the gut, these are playing on a sensitized organ, which can then be hyperexcitable and influence cortical perception of stuff, as well as having an end organ effect, which is amplified, because this then just keeps happening round and round, and you can then throw in some of the endocrine effects via the adrenal, which I haven't discussed today, which then are like the petrol on the fire. So here's your fire triggered by that spark, and here's the petrol that keeps it going. But there's a complexity to this which I want to persuade you of is real, and clever people are doing animal stuff to identify this, and simpler people like me are doing some of the human stuff just to identify what happens at sensitization level. But the clever stuff which I wanted to move into, the microbiome, before I sit down, is to say that a lot of the understanding of this is to think about how we've looked at this. Stress has become an interesting thing. So I don't want you to walk away from here thinking that everything that happens in your gut is stress, but stress is a model that allows us to understand how something acutely happening here influences stuff here. And if you have a sort of a handle on that, then it means that you can then think about how chronic diseases, not that they are stress, but they are, it shows the pathway that may be used. A chronic disease here may have an influence here, and then in turn going back. And the model is this. This is a little rodent, which has this stress of managing both a maze as well as being above water, which apparently is terrifying if you're a rat. So it has this water exposure stress plus a maze stress stroke learning. And it allows us to look at aspects of how that stress response can be fiddled with in terms of changing its gut bacteria, and also measuring pellet output, and with probes in the poor animal's bottom, we can assess how its gut works. So my research fellow wasn't doing too badly after all. He's got no maze to deal with. And the point about it is this. The point about it is that there is this gut-brain axis, which I've talked to endlessly now, is more complex than that. There is another player in that, which is undoubtedly present, which is the gut bacteria. Okay, this gut flora that you have, that has long since been formed, is an absolute third limb of this joint system. So from here to here, and within here, there's gut. And of course, remember that as far as the small bowel goes, we always talk about the small bowel being sterile, but it isn't. Okay, it's got about 10 to the power 5 bacteria per cl of fluid we take from it. It's not sterile by any manner of means. So any microbial, we tend to talk about chronic bacteria because there are literally trillions there, but there are also small bowel bacteria in health. So what this stuff tells us is that if you look at patients with various forms of psychiatric disease or psychological traits, there is an alteration of how they present with antibiotics. In other words, if you are somebody who's had courses of antibiotic, you're more likely to have psychiatric and psychological traits. Now, of course, that's a chicken and egg thing, is it that these patients are seeking help more? But it's a phenomenon that's occurring. And then can you look at causality from that data? You can then go back to our friend the mouse, and you can look at, when you give a mouse antibiotics, looking at how it behaves on that maze. You give a mouse an antibiotic, it becomes going through a maze. So you do something here, and it interferes with here. It's not just here to here, it's here backwards. That's interesting. And if you then put the water under that maze, it gets rid of it. So water plus antibiotics for this mouse. So here plus here makes all this terrible. Okay, this bidirectional thing is a big deal. And the microbiome is absolutely a key part of that. And that begins in early life. This is some different data from a very, very renowned group in Cork in Ireland, showing that if you give antibiotics either singly or in combination, and the earlier you do it, the more you interfere with the ability of this mouse to develop and learn. So you can alter the stuff here and stop this animal from learning. Some of these animals never learn how to walk through a maze because they've denied whatever it is that's happening magically here with bacteria, never allows this to develop appropriately in a rat. And there's then this classic thing of a germ-free model. You can take rat pups in particular, and mice also, and when they're born, not to separate them from their mother, but put them in a germ-free environment, essentially a vacuum, where they're absolutely not exposed to in their chow or in their water or in their environment. There's literally germ-free. And what you see is this. You see that these animals who are brought up in a germ-free environment have a very heightened stress response in terms of this pituitary axis I haven't talked about, I try to bypass, but they have an enormous stress response. Altering here also alters that petrol I mentioned in that fire. It doesn't just alter here, it also alters probably through a neural mechanism via the hypothalamus, but that's an interesting phenomenon. And that is modifiable. Okay, again, I'm not talking about treatments, and I've certainly got no axe to grind about any particular probiotic. But when you then give what this complex set of data shows, and I don't want to bore you with all this, but you can influence that learning response and the steroid response and the sensitization response by giving probiotic preparations in some animals. And whereas the sensory stuff is sympathetic nervous system, the adrenergic nervous system, the motor stuff seems to be strongly cholinergic. Remember I mentioned the IPANs, there's an upstream cholinergic contraction, a downstream nitric oxide relaxation. There is also the motility aspect is dependent on cholinergic mechanism. So TNBS is something which we use to induce inflammation in the gut. If you put TNBS compared to controls, you get lots of cytokines, these noxious chemicals released in the gut. That's what you'd expect. That's what the model does. But if you do that and you stress the animal, it really produces lots of noxious chemicals. You do all that and you add a drug which blocks the parasympathetic nervous system, and it all goes away. So this is the next phase of my revenge model, that there is this altered stuff that happens there which results in the comorbidities. So the belief that our patients with neurological disease who also happen to be depressed and so on, that there's a reason for that. There's the illness behavior stuff. There's also the potentially primary illness disease. So some people with lots of these neurological conditions get psychiatric disease. But as well as that, you end up with this chronic GI dysfunction, which has this kind of feedback effect. So you get the GI symptoms, the perpetuation of the psychiatric and psychological manifestations, and then the vulnerability to peripheral factors. And the vulnerability means it's also potentially treatable. So there's a role here for things that we may be able to do there. If you're vulnerable to something, that's bad news, but it also opens up the door to stuff that you may be able to treat. You can interfere the system here. That's what we've always done classically, but maybe we can interfere the system here in terms of microbes down there in the end. The part that becomes slightly more interesting still is then the effect of diet in all this. Okay, not to patronize you, but this is plainly an interesting area for all of us, and I'll go through this data in a little bit of detail if you don't mind. So this effect I've showed you there, just to remind you what I said before, my little nonsense about time, was that there's this model of disease which causes, where you get the disease, altered gut function, perpetuation of psychiatric disease, and this is a key part in what drives these symptoms being produced. This is the model here. So animals were trained and they developed this memory for working through that maze. And you see that there's this cognitive effects are the ones that relate to how good they were at doing stuff. And when they were given two different forms of diet, forgetting what they are for the time being, you can see that there's a difference in both their working memory and their past memory, so-called reference memory, of how they coped with the maze. So when you change the maze around and exposed it, turned it upside down or reversed it back to front, there's ability to look at so-called working memory as well as reference memory, which is influenced by diet. Okay, just talking earlier on today about, you can change how animals behave in a lab, something I haven't heard of before, by changing the chow that they're exposed to. This is the experimental version of that. And their food-seeking behavior also changed, so that when you were according to what diet they were exposed to, some of these animals, when you put chow in different regions of the maze, would stop or would start seeking food, not according to the amount of calories. The calories were matched, but according to the kind of way they were nourished. And these differences were related to not just the bacterial composition, but the composition, so I can tell you now, which differs about how much of it was protein in terms of calories and how much of it was just straightforward calories. So it's not an obsession about protein, but just so that that can drive memory and food-seeking behavior and even anxiety. Okay, so these animals which were protein-fed had generally higher levels of anxiety. So there's an interesting phenomenon. The detail isn't the key thing. I don't want you to walk out of here. I deliberately didn't overplay the protein or whatever else diet, because that isn't the point. The point is, because we don't know how this is going to translate from rats in a maze to humans in real life, but the point is more to do with the fact that this stuff, whether this may work through the microbiome, who knows, through altering the endocrine system, who knows, through a direct nerve effect, who knows, but it's there. And it's quantifiable for the first time. Once you have something quantifiable, we have ways of understanding it better. And that's what this is a really nice, sorry, the thing's fallen off the bottom. This is a really nice, readable review of this whole brain-gut microbiome stuff. The reference, it'll be on the slides, but it's called Nature Reviews in Gastroenterology and Hepatology, and it was from 2016, and the first author is Martini, as in the drink. And to come towards the close of all this, there are psychiatric diseases which are associated with abnormal genes. And guess what? Those genes that are abnormal in psychiatric patients are the ones which are not coding for any behavioral things that we know of, or they must do. They're coding for alterations in how your gut handles foreign microbes. Isn't that interesting? It may be completely phenomenological, but there are 2 genes identified in schizophrenia so far, both of which, looking at the immune system, both of which, one directly, one indirectly, has a question of how your gut signals responses to enteric antigens. And we know that, interestingly again, if you look at it a different way in terms of physiology, in schizophrenic patients, they have quite a lot of inflammatory change in their gut. There's 2 or 3 studies being done from North America showing that schizophrenic patients, unrelated to their medication, independently have altered peripheral nerve chemistry. Depression and other psychiatric diseases, not talking about just feeling mildly blue like all of us feel, but full-on major affective disorder, is associated and can be moderated by the microbiome. Lots of data to support that. And that in turn, if you look at people who are recurrent so-called cyclothymic, if you look at the microbiome in terms of stool analysis, admittedly, which is highly flawed, between the time when they are manic and when they are depressed, there is a difference. Causative, related to meals, just a casual thing, who knows? But 80% of people with major affective cyclothymia have an association and alteration of gut flora. I will stop there. I was going to finish off talking so much by just persuading you that actually the burden of gut, I'll say one thing with this one slide on there. The burden of gut disease for our patients is huge. This is spinal injury. It's huge for them, and apart from just being very prevalent, it also has a magnificently huge impact, and this is for MS, that of spinal injury. You can see, this is the population risks of these conditions. In MS patients, 2/3 have constipation, half are fecally incontinent, and fecally incontinent on a weekly basis. This is, soiling your pants on a weekly basis. That's a big deal. Okay? And also bear in mind another obsession of mine, 4% of us are fecally incontinent on a weekly basis. Okay, so look around the room. There's about 100 people here. You've got about 200 contacts on average, apparently. 8 of those people are incontinent on a weekly basis. This is a big deal in society. I'm obsessed by fecal incontinence. So it's hugely prevalent, but it's also a huge cause of distress for our patients. It's the thing that bothers them most. Okay, in MS patients, the thing that bothers them most, and in spinal injury, it's, you think it's all about not being able to get around. Actually, bowel dysfunction is hugely problematic for our patient groups, and you'll know that. I'm telling you stuff you already know, and it costs the system. Okay, we all suffer from this in the sense that not only do our patients, but we end up with, once you have bowel problems in MS or spinal injury, you're twice as likely to be hospitalized with non-bowel problems. So I don't want to go through any more clinical data because there's lots of it is phenomenological. I hope I persuade you there's a huge burden out there, that there is lots of communication between that, and that a lot of that stuff relates to the microbiome, which interferes with neural transmission through here, which interferes with through here, and may have an endocrine effect through that, but it's reciprocal, it's bidirectional. And if you're persuaded by even 10% of that, then that's been, for me, thus far worthwhile. And I will stop talking and ask you to start talking now, please. Thank you very much indeed. And who wants to start?